Hi, this is Eric Lyons, and this is a tutorial on how to use polymorphism tables as generated by the CoGe Comparative Genomics platform. These polymorphism tables are generated by scanning a multiple genome alignment and identifying every position where there is heterogeneity in terms of the character that's represented there. When such a polymorphism is identified, its position is recorded and a series of information is then retrieved and displayed within these tables. The information is displayed on the top of the table in this header row and includes things such as the position, a gene that may have been affected by the polymorphism, a breakdown of the individual strains and the characters that they have, as well as a series of links that allow you to go and use the CoGe system in order to further validate whether or not that polymorphism is real. One of the important things that you get are false positive scores, which allow you to evaluate, based on an automated heuristic algorithm, the likelihood of one of these polymorphisms being real. So, as we scan through this list, we can see that there's a variety of polymorphism types. The first one we come across is deletion that causes a frame shift, but it has a relatively high false positive score. It has a false positive score of 7. And over here is the actual polymorphisms itself. So we can see that we have a deletion in a series of Gs in different sequence strains. So if I click on this link right here that says show, this will show the alignment around this polymorphism by bringing in the 10 nucleotides upstream and downstream of it. So I can see that here's my polymorphism, and that it occurs in a series of Gs. Well, it turns out that for the technology used to sequence these genomes, which is 454, homopolymers are a source of sequencing error. It has a difficult time resolving a long series of the exact same nucleotide. So in that case, that's what's given rise to this false positive score of 7. So this one is probably not real. But I can scan through the table and I can see that I have a SNP with a false positive score of 0. And if I wanted to evaluate that in more detail, I can click show and I can see that I have a C to T transition. And if I wanted to evaluate that in more detail, I can use these links to GEVO, which will allow me to analyze the genomic region around this polymorphism in all of these sequence strains. So GEVO gives you a series of panels. Each one of these panels represents a genomic region. The dashed line is separating your top strand and bottom strand of DNA, and our polymorphism is this little spot. So if I scroll down, I can see that KSJ2 has this clear patch running through all these colored bars. These colored bars represent the regions of sequence similarity between pairs of these genomic regions. And so if I click on one of these, it will highlight which region it matches by drawing this transparent wedge. The blue line you see in the background represents my position within these genomes. So this is a spot that I've anchored myself in order to do these analyses. And this green tubular looking thing is one of the gene models. And we're only looking at 200 base pairs, 100 upstream and downstream from this particular polymorphism. So just scanning through all these, I don't see anything abnormal in terms of the local sequence around there. But what I want to do is I want to scan and look at a larger amount of sequence. So I can scan down this web page and I can specify using 1,000 nucleotides upstream and downstream from where we are anchored within these genomes, and in order to get a larger view. So from this larger view, I can see that here's the gene, here's the 3' end, here's the 5' end, it's on the minus strand, and here's my polymorphism. And I see that I have a second polymorphism in one of the other strains further away. And so I still don't see anything strange about the assembly or any other genomic feature, but I can click on these just to see if I get some maybe local regions of repeat sequences. But so far this polymorphism looks decent and putatively real. So I can expand this search by looking at 2,000 nucleotides upstream and downstream of that position and see if there's anything nearby. And as I scan through, I don't see anything anomalous, and I can just click these regions just to see if there's anything that might jump out. So as far as a polymorphism goes, there's nothing that I see within the assembly or within these alignments that may suggest something is wrong with it. So I would count this one as likely, and one that's probably worth following up with additional experiments. So particularly to clone and sequence this region, see if it really is real, and if it is, then move it into some genetic experiments. So next I want to show you one that has a couple of problems. This is one that I've identified previously and it's further down on the table. So I'm just going to scan down towards it. And it is this one right here. So this polymorphism is an insertion that causes a frame shift. And what I can see is I have an extra G and things look relatively okay within it. Actually, I'm looking at the wrong one. I need to skip up a little tiny bit and move up to this one here where I have a non-synonymous mutation. So I see another C to T transition. Everything around that region looks decent, and now I'm going to compare that again using GEVO. So GEVO again is going to anchor ourselves in these genomes and extract out by default with this link 100 nucleotides upstream and downstream from that polymorphism position. And immediately I see something suspicious, which is in one of the sequence strains, in this case KSJ11, I see a series of orange bars. And this represents ends, or unsequenced regions of the genome. These are probably spots where two contigs have been joined together, and that there's something that has messed up the local assembly around that region. I also see a lot of other polymorphisms around here, which makes me suspicious. So even though KSJ2 seems to have lined okay, or at least I have an assembly, there is hints that there's something wrong with these sequences in terms of causing problems with the assembly. So if I highlight this region, I see a different pattern than what I saw before. I see that there are lots of local regions of repeats where these things are matching itself multiple times in multiple areas. And I can see that that pattern is repeated among all these different regions. So whenever you have sequence that contains local repeats, this can severely impact the assembly algorithms because it's unable to accurately place each individual sequencing read, and you can get some errors. So I'm going to expand the analysis to look at a larger region. I'm going to jump straight out to 2,000 nucleotides upstream and downstream from this, just to sort of see what the overall region looks like. So there's one of the sequencing problems. KSJ2 sequenced through, or assembled through it fine, but I do have these gaps where I have assembly problems in these other regions. And when I highlight regions of sequence similarity, I can see that this spot here has lots and lots and lots of local repeats. The little regions are aligning to each other all over the place. And that this is really likely going to cause problems with assembly, and hence that this is going to be a polymorphism that I will consider to be a false positive. So I thank you for your attention, and I hope you have fun in using these tables in order to further classify and identify polymorphisms among your sequenced genomes.